So the task here is to look at the Ant Odometer stepping on stilts and stumps study that we're reading. We're looking at, on top of the independent variable, the dependent variable, the hypothesis, we're looking hard at the data. And we're trying to see if the data is conclusive by any means. And to do so, we've been looking at something called error bars in class. So, down to the data, let's see when they first start talking some data before we get to it. And when we do so, we're going to see that they say, let's just start reading together. These ants started determining homeward runs when they had covered the assumed distance to the nest. They switched to the nest searching behavior outlined above. So bottom line is we had some ants, they went to get their food, and then they basically found some way to come back. And they're testing the idea of how they came back. How did they use some kind of odometer, some kind of counting mechanism, possibly. That's what we're looking at here. Any case, the ants walking back homeward on stilts clearly overshot the home. Now, you got 15.30 meter. They give you this data. They give you the interquartile range to be 3.24 meters. Okay, the n equals 25. So we had 25 ants who were put on stilts. They glued some little taller pieces of, I guess, pig, I'm not sure exactly, segments on them, and they had longer, basically patterns of walking, so they clearly overshot that. So what's this data mean? Well, 15.30 meters, okay, is probably going to be the median, okay, and then the interquartile range, IQR is 3.4, is kind of like that error bar. So let's go to where we get that 5.3. So let's go to the data of the experiment to talk about that. And we'll talk about what that interquartile range is all about. So let's go to the data, and here it is. Okay, now, what are we talking about here? All right, well, we're talking about that 15 point something value they just gave me is right here. Okay, right here. Okay, 15. And they usually, when you're using interquartile ranges, medians are given. Okay, now this is called a box plot. And an interquartile range is something also known as a middle 50. So this is kind of like our standard error of the mean. It's not quite that, because this is a descriptive piece of statistics, kind of like standard deviation. This is showing me the spread that 50% of the population, okay, was in this area, okay? And what I mean by that is, let's go take a look at a distribution, and let me explain. So this is a lot like we've been doing, except this is not a standard error of the mean. This is a descriptive part, meaning it's not a variability. It is to a certain extent, and let me show you the difference. So let's start with a normal distribution, the best that I can draw. And what the central tendency they're using with a box plot, with those boxes you see, is they're giving that little dashed line as the median. Now, if it was a perfect distribution, your box plot would have the median right in the middle. See, what they're trying to say, if there is a normal distribution, the central tendency they're going to use is the median. So the median would be in the middle. Meaning if you were to measure the distances, this would be the distances, right, that the, let's say the tall or the stilted ants traveled. You can see that the middle number should be in the middle of your distribution. I know I didn't draw my curve perfectly, but you can get the idea. So not using the mean, they're using the median as their central tendency, and that dash in the box is showing where that median is. Now, what's the other parts of the box? What is these ends given? Because they give me something called an interquartile range, and that is, again, a weird piece of statistic that we haven't talked about, but we should be able to understand based on what we've been doing lately. So sometimes the interquartile range is called the middle 50. All right, now why is it the mid 50 or the middle 50? Well, because what they do is They say that, well, from this position to this position in again, a perfect distribution of data, this would represent what? 50% of all the population's distances. And this next half would represent the other 50%. So if you split this in quarters, and that's where the quartile comes from, you split this right here, you're going to have 25%. So what they do is they split the perfectly distributed curve here, and again, mine isn't drawn correctly, perfectly, but the best we can do, we can split the distribution of distances, okay, into quarters. And when you do so, we have the quartiles. So the interquartile, okay, value they're dealing with is in this range here. And sometimes it's called from the second to the third quartile. Or this is the first, okay, so the beginning of the second to this point. And who cares? The bottom line is sometimes they call it the middle 50 because we're dealing with everything in this range. So when we do standard deviation, we kind of do this, except we don't break it into quarters. We break it into 68% is the distribution of possible values of the organisms in the population have that value at plus or minus 1 standard deviation. And when you have 2 standard deviations, it's 95%. So they're just using a different way to report not reliability, okay, but variability. That's different. So variability is what we're dealing with here. Essentially, we're working with standard deviations if you want to deal with it. They're just using nice 50 or quarter percentiles. So the interquartile range is the range of the different distances on either side of the median of a perfectly what? A perfect distributed curve. And you're looking at the middle 50, okay, from the second to the third quartile, okay, if you want to look at it that way, or quarters. So this box right here that they're giving you represents this position. And this box, this end, represents this position. So these boxes are showing where 50%, okay, of all of the variable, where 50% of the population they're studied is where their distances lie. Okay, this would be the central tendency, in this case the median, okay, and this of course would give me the variability of the distances. Obviously, we want this box to be as what? Small as possible. So if we had a median and we're a really small box, this tells me that 50% of all the measurements I made on the different ants were very close together, and that's cool. But obviously you're going to not always get that. In this case, in this lab, you had n equals 20. You had 20 samples. And there was, what, 50% of all of the distances of each group is showing this range, this variability. Okay? Now, if you notice, they have these things that look like error bars, but these are whiskers. Okay? Now, what are whiskers? Well, whiskers can mean a couple different things, but in the lab that you're looking at, these whiskers show the 5% and the 95%, okay, percentage of ranges of where all the distances were measured. So if we went from 5%, let's get a better color here. Let's go with the, I don't know, red. I guess I could have picked that anyway. So if we go from 5% here to 95%, so this gives you a lot more possibilities. Okay, now of course this 5% to 95% is going to have not a lot of, hopefully not a lot of samples that had that distance, but it's going to show you how there could be a lot more, ones that are outside the norm. That's why these bars are not even. When we do a standard error of the mean, as we've talked about, the bars are even because we're dealing with plus or minus that same value. Okay, but with these whiskers, you may have a few more just by chance that are outside the norm, and your whisker could be longer. Let's go back to the study. So when you're interpreting your results, these are not quote unquote the error bars. You say, oh my gosh, these guys are overlapping. These are not standard error of the means. This represents 50% of the variability of all the distances. And this represents almost 100% from this line to this line. This is from 5% to 95%. And if you read carefully, they tell you that below. Okay? Box plots show median values. So that's the median value of the homing distance when n equals 25. I might have said 20, so I have 25 ants per experiment. As well as the interquartile ranges. Okay? The box margins that we just talked about. And the 5th and 95 percentiles, which are the whiskers. So these whiskers tell us where every single measurement lied. Of course, the bigger whisker means that there was some way outside the norm. Okay, that's why, again, these are not standard error of the means. So when I'm looking at box plots, I'm treating this range kind of like the standard error of the mean. 50% of my measurements are here. 50% of my measurements are here. Okay, so you can see if there's tremendous overlap, let's say from here to here, there's less significance. Okay, so something to think about when you look at this. This is variability. It's not reliability. It's not a standard error of the mean, but you can look at it in terms of significance. Is these guys, the stumps, significant compared to the normal, the control? Okay, and the answer is yes. Their variability total is here, and their variability kind of overlaps. But remember, this is a whisker. This shows you just a whisker is a small little thing on an animal. Yes, it's important, but this whisker tells me there's a couple of points way outside the norm. But within 50% of all my distances, we're right here next to the mean. So the mean is much lower than the control, and you can see that this range of 50% is not overlapping this 50%. So something to think about when you're interpreting this data, and I want you to understand we're not dealing with standard error of the means, we're dealing with variability. More like you're dealing with a standard deviation. Hope that helps.